these networks in the spinal cords as these nonlinear elements, and we're able to show that you could have just one circuit of these neurons produce these different patterns. So by changing the energy or the signal from the brain, you could have your horse go from a walk to a trot to a canter to a gallop. And this challenged the notion in neuroscience. So the idea in neuroscience community at the time was, no, you actually don't have one circuit, you have 4 different circuits. So when the horse wants to make the shift, the horse is going to jump between these neurons. We says, no, you don't. You can just have one wired circuit, tune it up or tune it down, and the animal can make the jumps.
By tune it up or tune it down?
Increase the energy or signals that are coming in. So it might be that you increase the amplitude of the signal, the size, or the frequency of the pulses that you deliver.
Okay.
And the application here is really getting after leg robots. So there's a lot of interest increasing in industry and in the Defense Department in creating leg robots that can be sent into nasty areas. So could you send leg robots onto a beach to demine the beach before you send in your troops? Could you send it into dangerous areas like a nuclear power plant that had an accident to go and decontaminate the area? Can you do it for surveillance in areas that are under attack?
The idea of controlling these robots is the idea?
Exactly. So you could use much simpler circuits based on some of the work that Ian and I developed to now control the legs of the robots. And there are several labs, primarily labs in Japan, that have actually been now incorporated these circuits.
Stochastic resonance. What is that?
So stochastic resonance is the physicist term for the phenomena we spoke about toward the beginning of the interview, and that's this counterintuitive notion that you can put noise into a nonlinear system and enhance its ability to detect a weak signal.
Okay.
And it's a phenomena that was actually brought in to account for the periodic recurrences of the Earth's ice ages. So the Earth has an ice age about every 100,000 years. We're overdue for our next one. And in the 1980s, early 1980s, 2 groups of theoretical physicists in Italy looked at these models and came up with this idea of stochastic resonance, and that is a little bit of noise could help switch the Earth from an ice age and a non-ice age state. Our lab, starting in the mid-90s, recognized that you could use this phenomena to tune up or tune down sensory function in humans. And so we've applied it to the balance control that I mentioned, but we've also applied it to touch sensation. And a condition that we've been quite interested in is diabetes. So with diabetic patients, they often can develop peripheral neuropathy, where as a result of the disease, they lose sensation in their fingers, their hands, and their feet, which can pose substantial problems. You can imagine now that if you can't sense what's going on at your foot, if you have a pebble in your shoe, you don't know that there's a pebble in the shoe. That could lead to a cut on your foot. If you can't feel that, you don't know there's a cut. In the worst case, it could lead to an infection, which could develop to the point where you might have to amputate your foot. And most think, oh, this could never happen. But in the United States alone, there are 50,000 to 80,000 amputations due to diabetic neuropathy each year. What we've been able to do with stochastic resonance is develop devices in addition to the insoles and the stroke rehab, noisy socks, for example, that can be used for diabetics to enhance their ability to feel and thereby substantially reduce the risk of pressure ulcers on their feet. And we can lower the detection thresholds close to the levels of young people.
Okay. Now, you've also done work in genetic engineering.
Yeah.
Now, quite a range you have here. How does this relate? Does any of that work relate to some of this, or what's involved there? Is it a totally different area?
It's not totally different. So we made the shift in probably the late 90s. We, like many engineers, it kind of felt left out from the genetic engineering revolution. None of us really viewed that as engineering. The idea of kind of bringing in a gene from one organism into another, to us, was no more engineering than changing a red light bulb to a green light bulb. And we similarly, in the biomedical engineering community, were kind of looking in, like kids outside a candy store, at all that was going on in the genomics revolution. As there was a lot of attention, much of the funding was going for sequencing genomes. In the late 90s, we got encouraged to think about what could engineers do in molecular biology. And we got excited in thinking about the large kind of sets of components that were coming out of the sequencing efforts in terms of genes, proteins, promoters, and realized as engineers, boy, this could be a fun toy box for us, similar to the erector sets or circuit electronic sets that we all had as kids, or radio sets, and realized, hey, as engineers, we could take these genes and proteins and put them together in now biological circuits instead of electronic circuits in new ways to create interesting circuits that could now be put into cells to reprogram the cells.
Substitutive genetic engineering?
It would be substitutive genetic engineering, really a field now called synthetic biology,
okay,
where you're going to design synthetic gene networks, for example, out of biological components. So for example, in one case that we did, based in part on nonlinear dynamics, we developed what's called the genetic toggle switch. So a toggle switch is typically found in electrical engineering. It's a switch that's at the heart of all of our computers. So in your BlackBerry or PC at home, they are simple memory elements. So your computer will have thousands, if not millions, of these switches. We thought about how could you make one of these switches out of genes and put it into a cell. And with Tim Gardner, we realized, okay, you could do it if you could take two genes. If, say, you have gene A and gene B. If you drive them with promoters, which are their on switches, so that each wants to be on.
Yeah.
Now if you set it up so that the protein produced by the gene wants to bind to the on switch, let's say the protein produced by gene A wants to bind to the on switch for gene B, shutting it off. If you then take the protein produced by gene B and design it so that it wants to bind to the on switch for gene A, you now have a system where gene A and gene B both want to be on, but they want to shut each other off. So in principle, you can design this as a so-called bistable switch, so that it wants to exist in one of two states. State one is gene A is on, gene B is off.
Yeah.
And state two, gene B is on, gene A is off. And so you can now toggle it between those by just delivering a little bit of a chemical pulse to shut off the currently active gene.
Okay.
Which now will allow the gene that has been kept off to come on, and it can now shut off the other guy. And this opens up a lot of applications in cell therapy and gene therapy, where you want to introduce genes or proteins that are missing or have been damaged in the patient, but you want to regulate how they're producing the protein of interest.
So it really calibrates or
Absolutely. And it's going to be kind of a safety switch.
Can you give me an example of where specifically this is being worked on, in what diseases?
So we are primarily right now actually using it to explore the genes underlying stem cell differentiation. So we can now go in and flip on genes at very critical points in the development to begin to understand which are the genes that need to be turned on or off to develop to basically take, say, an embryonic stem cell or a pluripotent, a reprogrammed adult stem cell to now create heart cells or muscle cells or bone cells.
So that sounds extraordinary. Had there been other ways in which to grow these?
There had been other ways to switch genes, and the contribution that we made was that these others were really what are called pressure switches. So these are switches that needed to have a chemical present to keep the gene on or off that you're trying to control. So the idea would be if you come into your studio here and to keep the lights on, you would have to have your finger always on the switch. The challenge when you want to look at biology is that if you now have this extra chemical, it can really confound your results.
Right.
Because your cell and all that's in it could be responding to those chemicals. In our case, we have this toggle switch, which now you only have to deliver the chemical once to flip it between the on or off state. So it's like a typical light switch in your studio, where you come in, you flip it, it's now on, you don't have to worry about it, and then you can just flip it off.
I want to talk a little bit about your background as a scientist. Your training is in physics. I mean, you're talking about yourself as an engineer, and I guess that came later.
Right.
But as an undergraduate, you got your Bachelor of Science at Holy Cross in physics. And then you won a Rhodes Scholarship, and you went to Oxford,
right,
where you did bioengineering.
That's right.
Is that right? Biomedical engineering. That's where you got your PhD, which I think is interesting because you got your PhD in England.
Right.
I don't know how common this is for someone going on a Rhodes Scholarship. I mean, often they will do a master's and come back.
That's
but
right.
as a scientist, getting your PhD in England, do you think that that had some bearing on the way you think, on the way you look at science, being abroad, training here, going abroad, and then coming home again?
Yeah.
Has that shaped you as a thinker?
I think it has, and I think maybe even stepping back, I'll comment a bit also on the role of Holy Cross. So Holy Cross is a liberal arts college, a small undergraduate college, and I actually have a BA in physics. And
you have a BA, not a BS. Okay.
Not a BS, and I was brought up in a family of engineers and technical engineers. So my dad and my uncles were engineers, my mom's studied mathematics and taught mathematics. And I thought I was going to study engineering. Okay. And I visited engineering schools and found that nobody was smiling, everybody kind of had their head down, and there was a lot of oil smells in the hallway. And when I went to Holy Cross, people were smiling. And for me, it came down to MIT or Holy Cross. And in looking at it, I realized that at Holy Cross, I could get a really solid education in the classroom in the sciences, and quite broadly, but that I could also get an education outside the sciences by interacting with many non-scientists on campus. And I found the same at Oxford. So I was strongly discouraged from going to Oxford to do my graduate studies by my scientific mentors. They said two things. One, Oxford isn't known for science. It's Cambridge. That's the university to go to in England. And two is that you'll be outside the community of scientists when you come back to America, and it'll be quite hard. But what I found at Oxford, which I haven't found so much in American universities, is that there's this incredibly active, almost exhausting intellectual life that is broad. And so in the middle common rooms in the Oxford colleges, I rarely was talking with scientists. I was talking with people studying economics or literature, philosophy. And I think it really helped, on the one hand, kind of broaden my view on what were interesting problems, but also gave me an opportunity to explain sorts of things that I was doing to the non-scientists. You know, on the former, it, I think, also gave me the base, both Holy Cross and Oxford, a broad base that allowed me to jump between these fields. The traditional route in America is you kind of pick your system as a grad student and a postdoc. You get trained by your mentor and you become an apprentice, and you study that system for 50 years.
Right.
And I haven't done anything close to that.
Yeah, I mean,
we've been able to
you have jumped
between
around.
areas.
I mean, you're both, in a sense, you are a scientific popularizer on some level.
Right.
You are a serious scientist, you know,
Right.
a laboratory scientist. You're also an entrepreneur,
Right.
from what I can make
Right.
out and what you've told me. And you've worked in so many different areas,
Yeah.
too, in terms of the research that you're doing. So
Yeah.
there is that broad base. And I guess the perspective that you got, or as a somebody starting out in a liberal arts school and then going abroad, maybe having the perspective from across the ocean.
That's right.
Gave you that global
I think it gave
view.
it gave me the global view, and I think also helped feed the efforts that we do in our lab on being creative, in that by actually being exposed to so many different areas, in particular fundamental scientific areas, we've been able to see connections between different areas that have actually afforded us to make some important contributions as we step into new fields. So we've also found that I get bored easily, and it's easy to kind of see when you're not the expert, to come in as kind of an informed amateur of where there might be connections that the experts, the ones who kind of really in on the details, haven't seen.
I see. Well, you're also, and it doesn't surprise me at all now meeting you, you're a terrific teacher. You've won awards as a teacher. I wonder what do you see in students nowadays, especially students in science? You know, there's been a lot of lamentation about the fact that we're not competitive and this and that, but I wonder if you have a different perspective. What do you see as the potential in our students? I mean, you are teaching at Boston University. You teach undergraduates and you teach graduate students.
Right.
I'm specifically interested in the undergraduates.
Yeah, I primarily focused on undergrad teaching.
Okay.
I just finished our semester where I had 100 of our students for one of our core classes of control theory. And what I see is that our students are hungry for relevance for the material. So we see the biomedical engineering students, and that they want to be engaged in the material. They want to see why should they be studying this. I don't find that they're critically focused on salaries. I think they would like to make an impact. I think the students today tend to be overwhelmed. I think one of the downsides of everything being hyper-networked is that they're being faced with way too many choices, so much so that they can't actually find the material that they need to find.
Okay, well, I think you must be an inspiring teacher.
Oh, thanks.
I want to thank you so much for being here today,
it's a
James
pleasure.
Collins. And thank you for joining us today for the Drexel interview.